we began by thinking anew about how you would do CNS-related research, so central nervous system research for diseases. The field is plagued by this common problem. You go into a clinic, you see certain observations in thousands of patients, you say, oh, well, if I could suppress that observation, I should get a drug that works, and it's been 100% failure in this field for the last 50 years in Alzheimer's and Parkinson's and ALS, in Huntington's, et cetera. And they're all characterized by a protein, which we call a misfolded protein, in the Parkinson case, α-synuclein. And in Alzheimer's, et cetera, it's often a protein called tau or beta amyloid. What we've seen is that these aggregates or misfolded proteins that are observed in the disease emerge. They're characteristic of the disease, but they don't actually cause the disease itself. And that observation that we made that led us down the road of identifying c-Abl as what we think is the most important target for disease modification came about from the fact that we could reproduce the entire disease process pretty faithfully in mice. We're using mice because we have to study the disease process at a rate of disease progression that mimics what happens in humans. So in humans, that's 1/3 of a lifespan, or 25 years. In mice, 1/3 of a lifespan is 1 year. So we can do experiments that are very hard to do, but we can do experiments for 1 year in hundreds of mice at the same time. What we cannot do is do similar experiments like this in monkeys for 10 years before they show a disease. So that's why we chose mice to do this work, we and a whole lot of collaborators at many universities worldwide. And so what came about was the observation that we could introduce synuclein aggregates through genetic or by just injecting protein aggregates themselves into the parts of the brain or the parts of other areas in the body, such as the GI tract, and then just wait and see that the disease develops. Once we figured out how to do this, it develops at the same rate relative to lifespan that humans do, about 1/3 of a lifespan. When we knocked out Abl conditionally in the brain, all of a sudden, all the neurodegeneration disappeared. So when we saw that, we knew that we had come upon an important regulatory factor in the disease process. Those studies led over a period of time to identification of all the downstream effectors that give rise to all of the manifestations of the disease that we know. At the top of that cascade was Abl, this enzyme. Abl's role in biology is to act as kind of a sentinel for damage or defects. And so what Abl does in response for the presence of these misfolded proteins is that it sees them when they enter the neurons. And when it sees them, it acts to kill those neurons. That's its job. So we wanted to block the ability of Abl to recognize the presence of these misfolded proteins and kill the neurons. Surprisingly, what we found is that when we add an Abl inhibitor like 1489, which we did in animals that had significant disease for a long period of time, something equivalent to a 10-year disease point in a human, we found that we didn't just block Abl and suppress what happened in those neurons, we actually reversed what had already occurred. So while we've called it neurodegeneration, you don't actually lose the neurons. They're dysfunctional, but they're not gone. When we blocked Abl, we saw that we could protect neurons from degradation. And when we protect neurons from degradation, we restored motor function, which we could measure how mice walk across steps, or how mice descend poles, or how mice grab a metal grate, and we can measure how hard it can pull on a grate because we can use a gauge to measure that. And we saw recovery of that function, and then we went and looked inside the tissue to see what else occurred. Surprisingly, Abl inhibition didn't just restore function and protect neurons from degrading, it also cleared the aggregates from the animal. And that's been the kind of holy grail. We only see those aggregates in the disease state, and now we had a mechanism to not only restore function and protect neurons from being lost, but we could clear the underlying pathology. So it's not really a cure, we can't say that, but it heads down a path where cure might be possible for some of these diseases. So in our case, it's been really eye-opening. So we moved our whole company in this direction about 4 years ago. Fortunately, got a lot of support from the National Institutes of Health, from the Fox Foundation, et cetera. Went public a year ago. Then the FDA has been very supportive. We went from no data in the clinic all the way into phase 2 in 15, 16 months. And now we're in phase 2 trial. So very, very quickly.